Okay, so first of all, I'd like to thank you very much, the Association El Argentina and all the Spanish countries to invite me for this conference on iron in amyotrophic lateral sclerosis. Is it a clue or not? Well, I will go fast on it, but you know that amyotrophic lateral sclerosis has a very high incidence, but a high rate of death, and it's characterized by neurodegeneration, mainly, but not only, of the motor neurons, leading to paralysis. But we also have a lot of hidden behavior and non-motor symptoms in ALS. This is the clinical description. The physiopathology also associated TDP-43 deposit, for which many studies have to be done to see whether it's associated with inflammation, iron, aggregation. But what we know is that from many years, we know that there's an atrophy of all the motor system in the brain, in the brainstem, in the spinal cord. And before this atrophy, we have an iron accumulation. This iron accumulation can be measured by MRI with high field Tesla, so 3 Tesla, for instance. And we know that with different type, notably the quantitative susceptibility measure, which is a new measure, post-treatment measure of iron in the brain and in the spinal cord, we can observe that indeed there's an iron accumulation in different areas of the central nervous system. It has not been fully demonstrated, but in the preclinical setting, we observe that before having atrophy, so before the neuronal death, we have a slight iron accumulation. And with the propagation, you have iron accumulation and then degeneration of the motor neurons and the motor neurons. And you can see here, for example, in one publication, the different areas where you have an increase of iron. So it's very, you can have basal ganglia, substantia nigra, and some part also the cortical tracts, obviously. So we also have other proof showing that there's an iron dyshomeostasis in ALS. If you measure iron in the cortex or in the spinal cord, you also can measure with mass spectrometry, for instance, that there is an iron accumulation in the area of the central nervous system, which degenerates. And we also know that the ferritin level is increased in ALS people as compared with control. And this can be also associated with shorter survival time. So the highest level of ferritin, the shorter survival time. Well, if we say that, we need to bear in mind that 30% of biochemistry is using iron. So in fact, when you say iron is involved in ALS, you can also say iron is involved in Alzheimer's disease, in cancer, in inflammation, in everything, because 30% of biochemistry is using iron. Life on Earth appears notably thanks to iron. Why? Because iron is able to give and is a donor of electron. So iron is involved in mitochondrial respiration, in neurotransmitter synthesis, in myelin synthesis, in NO metabolism, etc., etc. Interestingly, the loss of iron is even more dangerous than the iron accumulation. So you really have to have a very tiny regulation of iron in order to avoid degeneration. For instance, if you use neonate mice for which you reduce the level of iron in the diet or you increase the level of iron with diet, then you can worsen degeneration at the adult age. And also, it has been shown, notably in Parkinson's disease, that the area of substantia nigra, which is very highly active, is the area with the highest level of oxygen consumption and this area is degenerating. So it's not by chance that the motor neurons, which is the highest rate of oxygen consumers in the body, is very vulnerable and is involved with iron metabolism. So in 2012, there was a big discovery, because we believed before 2012 that there's only a few types of regulated cell deaths, notably apoptosis, which is for programmed cell death, homeostasis with embryogenic life, autophagy, and necrosis. But a number of regulated necroptosis has been described, notably ferroptosis. And ferroptosis is a new type of iron-dependent cell death. Indeed, it is characterized by an iron up-regulation, and by doing this, it can lead to lipid peroxidation, which degrades the membrane. And what is really interesting with this ferroptosis, which has been described first in the breast cancer, that numerous key actors you can regulate to avoid this ferroptosis. So this is a new highway of different pharmacological target to reduce this iron-related cell death. But now the question is whether or not this ferroptosis occur in ALS. In fact, in this system, it's a little complex, but let's make it simple. You have importation of cysteine in order to have glutathione, exportation of glutamate, and we know that we have an excitotoxicity of glutamate in ALS. You have a lower level of glutathione, and glutathione is a cofactor of GPX4, the glutathione peroxidase 4. And this enzyme is a key enzyme for survival. If you don't have GPX4, you can't survive. Why? Because there's a detoxification of lipid peroxidation. You reduce the peroxidized lipids in lipid alcohol. So this enzyme is the key. And when you make a conditional KO of GPX4, you induce motor neuron degeneration. And it's very fast, very strong, and this has been demonstrated. I will go through different preclinical results, but when you measure the GPX4 protein expression, it is largely reduced in people having familial amyotrophic lateral sclerosis, sporadic amyotrophic lateral sclerosis, or SOD1 mutation as well. And this is true for people having ALS, dying from ALS, but also in the different models of SOD1, the different model of SC9 or F72. And when you now overexpress the GPX4, you delay the weight loss, and you improve locomotion and survival is in SOD1 G93A mice, meaning that GPX4 is really important for the motor neuron survival. Importantly, in the picture of ferroptosis, there's another key enzyme, LOX15. And LOX15 is also an enzyme that is directly dedicated to reduce lipid peroxidation. And we know there's a very large amount of lipid peroxidation in ALS. It has been published, notably in neurology, that you have a very high level in the blood of 4-hydroxynonenal, for instance. And when you block this enzyme, you can protect all the mice model of ALS from motor neuron degeneration. Interestingly, an inhibitor of LOX15 is currently in a clinical trial. So in the definition of ferroptosis done by the group of Stockwell in Colombia in 2012, they said that this type of cell death can be counteracted by iron chelation. If you reduce iron, you reduce this ferroptosis cell death, the ferroptotic cell death. So that's exactly what we have done. And we use deferipron as a prototype, and we use different doses, and we demonstrate that at 50 milligrams per kilogram per day, we were able to increase the survival of SOD1 G96R. It is the most severe SOD1 models. And in male, we create a higher dose to also reduce the survival. So thanks to this preclinical data, we moved to a clinical trial. And we first did a pilot clinical trial of safety in 23 early ALS people, already all under Riluzole. And we follow them for 3 months without treatment, and then 3 months with deferriprone and 9 extra months of deferriprone. It was open label just to check that the safety profile was good in order to move to a larger randomized double-blind placebo clinical trial. And ALSFRS, so the functional handicap and the iron level in the brain were the main outcome. We also compare with the natural history of a large cohort of 512 people from the MitoTarget Trophos trial. What we observed in people having ALS is that before deferriprone, they lose 5 points during the 3 months of survey, and it was only 3 points under deferriprone during the 3 months following the treatment's onset. And when you look at the MitoTarget on the 12 months, they lose 10 points, and on the 12 months, people under deferriprone lose only 6 points. So it was very encouraging, but we need to be careful. It was in an open label. And so we also observe that they're starting to lose less weight, which is also a good sign of disease progression. Interestingly, we measure the iron content in the brain and in the spinal cord of people in this trial. You observe here in the black the progression of iron in the spinal cord, in the cerebellum, medulla oblongata, and the precentral cortex. And you see there's a trend, non-significant, of increase. And there's a large reduction here in red under deferriprone in the 3 first months. And it goes to a certain limit. And after that, there's no more reduction. And the ferritin transiently reduce and then re-increase with disease progression. So we published this data with the preclinical data and the open label data. And then we obtain French funds to lead a large clinical trial called FAIR-ALS-2. FAIR, it's focal accumulation of iron. And we choose a dose of 30 milligrams per kilogram per day. Why? Because the same dose has been tried in people having Parkinson's disease in a pilot trial that I've led, and it was well tolerated, and it looks like being positive on the progression of people having Parkinson's disease. But for people having Parkinson's disease, it was more complex. Why? Because you need to treat people with levodopa, because iron chelation, as I said, iron is involved in neurotransmitter synthesis, and notably, iron is a cofactor of tyrosine hydroxylase. So if you put deferriprone in people having Parkinson's disease, you can reduce dopamine synthesis. So you need to co-prescribe deferriprone with levodopa in Parkinson's disease. So we just published in New England Journal of Medicine that a treatment with deferriprone at 30 milligrams per kilogram per day in de novo people with Parkinson's disease worsens the situation if you don't co-prescribe levodopa. But in people with ALS, because they apparently do not have a huge dopamine depletion, this should not occur. So the design of the trial is a very easy one. It's a 2 parallel group. You see on 48 weeks, end of the treatment. So it's you have, and then the ALSFRS with, we call it the combined ISFRS and survival score at 1 year. And now all the people have been recruited. This is an old newsletter. So we have 184 people with ALS that have been recruited in all these French centers. And the last patient will finish in December. So hopefully we will have the data in the early 2024 to see whether we can reduce the progression of LSFRS with deferriprone at the dose of 30 milligram per kilogram per day, split in 2 doses, morning and evening. Ferroptosis also is an interesting field because, as I said, it's a very new type of regulated cell death with key players on which we may develop pharmacological target, but also it could also help developing biomarkers. As you notice, we can measure iron with MRI, and this could be a biomarker to monitor whether we can reduce the iron accumulation, and this may protect against ferroptosis. But we can also measure iron through ferritin, as I said, and we published that there are 4 markers. If you combine ferritin for iron homeostasis, if you combine 4-hydroxynonenal for lipid peroxidation in the blood, and also NFL, so the light chain neurofilament, and 8-oxodesoxyguanosine, which is a DNA degradation, combining these 4 markers, you can predict whether the patient will be a slow or an intermediate or a fast progressor. So we believe that, and this is our hypothesis, that by stratifying the patient, having the highest dose of 4-HNE, combining with an NFL, 8-oxodesoxyguanosine, and ferritin, might be people that may respond to iron chelator and may respond to antiferroptotic drugs. And we can monitor this progression thanks to this biomarker. So I would like to finish with a takeaway message for iron. So this new type of iron chelation, we called it conservative iron chelation. Why? Because we don't want to chelate iron out of the body. We want to remove iron from the area with overload, or the overloaded area, to other areas that needs to use iron. So it's a redistribution of iron. That's why we call it conservative iron chelation. Ferroptosis could be the missing part to explain this relationship of iron accumulation and cell death. And ferroptosis could explain also the efficacy of conservative iron chelation, and could also pave the road of multiple other drug candidates to associate with a combined therapeutic approach. And this could also be part of a panel of prognosis biomarkers. So I'd like to thank you very much on this, and I would like to finish on the fact that there are many, many other drugs that are currently under development in ALS, hoping to find new drugs in combination very soon. Thank you very much.
Thank you, Dr. Devos. That was really interesting. If you want, you can stop sharing your screen now, so we can have a little chat here. Thank you so much for that presentation. It was very interesting, and it's certainly a very innovative and new approach. All these iron strategies are not one of those that you heard much about during conversations around the world. So that is why we wanted to talk to you, because I think it's interesting and it's important to also have a look at these new strategies related to iron. And so again, thank you for that. And just to make a recap, of what we just saw, we can say that okay, the Ferripron trial, the FAIR ALS trial, is on a phase three context, in this moment, on a phase three environment. So are we expecting results for the first months of next year? And the second question would be, are those results more likely to be definitive?
That's a key question for people having ALS. It's more a phase 2 than a phase 3 because it's investigator driven. I took the lead of this trial, and I asked to Apo Pharma, the former company which holds Deferipron, to be partner on this. And then Apo Pharma has been bought by Chiesi. And Chiesi, so now Chiesi, it's an Italian pharmaceutical company. Chiesi holds Deferipron. But Deferipron is out of date, as an out of date patent. So we will see what will be the result. If the results are negative or not good, probably this will be the end of the Deferipron. But maybe other new iron chelator or other new anti-ferroptotic drugs will be developed. As I said, there are already another one, LOX15 inhibitor, which is currently in the clinical trial. But if the trial Deferipron is positive, then there will be discussion with Chiesi whether or not they can move forward to another phase 3 trial. So probably it will be like, like with Amylyx Lab, with Tutka. When they did a phase 2 trial, it was positive, not unexpectedly, but it was positive. And then they have to perform the phase 3 in order to have registration everywhere. Even if FDA and also the Canadian FDA said, well, okay for, giving it to people, but still you need further proof for a definitive registration. So yes, we will have the result in 2024. I will fight to develop it if it's working, of course. It will be also bring new pharma company with other iron chelation in the field, because there's already new iron chelator in development. So whether it will be Deferipron or another molecule, we don't know. But hopefully Deferipron.
Okay, clear. Thank you. So 2 new drugs have been approved in North America in the last year or so, which are tofersen, as you know, and AMX0035. They were approved in North America, but not in Europe yet. So what is your stance on these new approvals?
Tofersen is very, very specific because it's, it's oligoantinucleotide. It's very specific for SOD1 mutation. So it's only for a very small amount of people, treating very early. So it makes sense to support this development for a very small part of the population. But as you know, so far, the clinical results are not strong. But we all believe that it's a good way to follow. For MX 0035, the results are real. You can't say the reverse. I mean, the results are real and quite strong, even a little stronger than riluzole. It's not a huge effect, but it's a real effect. And I think everybody believes in this effect in this trial. But if you go back several years ago, there's also a study with dexpramipexone that has been done by Merit Kutkovich with a beautiful phase 2 trial published in Nature Medicine And then the large phase 3 was totally negative. So we need to be cautious. And I can say that because, as I told you briefly during the presentation, I did a trial that I just published in New England in which I worsened the situation in people receiving deferiprone because they did not have levodopa. And I will publish another paper in Lancet Neurology soon, showing that if you combine levodopa with deferiprone, then you can improve people. So we need to be very cautious, because if you don't enroll the good people at the good time with a good dose, we can also worsen them. So for Amyx 35, there's a trial now, Phoenix. Currently, it will be finished in a few months. It's a question of months. As soon as the result will be published, EMA and all the European drug bodies will accept it. So, but it's just a question of being cautious. And it's also different. I don't know how it is in Argentina, but for instance, in Europe, in most of the European country, and notably in France, if you register as a drug, then you give a price of drug, and then you can prescribe to everyone. So it's not like in Canada or in US. They said, well, you can prescribe it, but I don't pay for it. You go to the insurance, and then you deal with insurance. So it's not the same spirit in France, for instance. Well, we said, is it working? Yes or no? If it's working, it has to be for everyone. So, and not just for the rich people that can pay for it. So it's different mindsets. So that's why in Europe, and I know well because I was involved at the European level, everybody said it's really promising. We believe it's working, but it has to have the phase 3 trial published in order to release and to go for the registration and to give a good price. I know it's hard when you're a patient to hear that, because you say, I don't have months in front of me to have this treatment. And I can tell you something. I was in a jury, and some ALS people were with us dealing with when we will have the treatment. So we need to go fast. And so we asked to EMA to go faster on the registration once the result will be delivered. And I think they will do it. So cautious, because we are several studies, several examples for which the previous trial was positive and then it was totally negative. So no clear benefit for patients.
That was very clear. Thank you so much for that. Are we having a similar situation in Europe with oral edaravone, or the situation is a little bit different?
Yeah, you are right. You know very well all the dossier. In fact, yes, it is different with the edaravone. Well, the spirit with Amylyx and with edaravone was the same. They tackle people very early. It was a more homogeneous population, not to advance in terms of LSFRS score, and so it was clever. And I think this is the best population to respond well to a treatment. So we've learned a lot with the trial of edaravone, because the first phase 3 was negative, and then they look at the subpopulation in which they can respond. And once again, I believe what they said was true. I mean, in this population, the people can answer better, because when they are too advanced, the LSFRS score can no longer measure the benefits. So that's why it's clever to tackle an early population like they did. But the intravenous administration of edaravone show clear benefit in this subpopulation. And the European Medicine Agency said the same. They said, well, it looks like being positive, but you have to do a large phase 3 to prove it. And they refuse it. It's not like Amyx 35, because they said, no, we don't do it, because, and why they didn't do it? Because it was a heavy burden for people to go back to hospital all the time for intravenous administration, only for very weak effect. And if you are a patient and then you feel months after months, the degradation, and still you have to go to the hospital for intravenous. At one time, you said, well, I'm fed up with this. I don't want anymore. Leave me alone with this. And that makes a lot of sense. So they were clever to say, well, let's move to an oral form of edaravone. So that's why they didn't perform the large phase 3 with intravenous administration, but they moved to an oral formulation. So now we wait for the result of the oral formulation. And I'm not so confident because like Amylyx, I'm very confident, but for edaravone, I'm less confident because the effect is really weak with intravenous administration. So I hope that we would not lose some effect with the oral formulation and then becoming no more significant. That would be a drama.
Yes, thank you. Lastly, we can expect, and we are praying, of course, for the ferriprone to be approved at some point, to be successful, to show an effect. If that is the case, so it will combine with the other drugs, or we can imagine a kind of a combination treatment, a cocktail treatment, right? You said that in this trial you've been using subjects that were on riluzole. So how the target mechanism of the ferriprone or the iron chelation treatment will somehow combine with the rest of the mechanisms that are targeted by the other drugs?
Yeah, that's a very good question. Yes, we all believe that the future of the treatment of ALS will be a combined therapy with many different drugs, like for HIV, like for cancer. We all know that, and everybody believes it. So already we know that for edaravone, Amylyx, or even the ferriprone for the first trial, it was already combined with riluzole. So it was the effect plus riluzole. Now the question, and we discuss a lot with the board at the board of pharma company when we discuss the design of the trial, whether we need to combine edaravone or Amylyx in the next coming trial. And so we decided, yes, we need to combine all the drug available, and then we will make a stratification for the statistical analysis to see whether there's an improvement as compared with the other treatment already prescribed. So if the ferriprone the result is positive, and if I can convince KSC to do a large phase 3 trial for registration, it will be definitely in association with the treatment already on the market, hopefully Amylyx, maybe edaravone. Then, is it enough? Probably not. Are there synergistic action? Yes, because the iron chelator is reducing slightly the iron content in the cells and the mitochondria, reducing the reactive oxygen species, so oxidative stress, and blocking ferroptosis. And if you are, for instance, if you're using Amylyx, it's also reducing by other way the oxidative stress. So there's like a synergistic effect on this level. If you use riluzole, then you're acting on the glutamate, and we know that there's also an excitotoxicity, glutamatergic excitotoxicity. So you are combining by different way to try to protect the motor neurons.
Great. So hopefully we will have some news next year. So once again, Dr. Devos, thank you so much for taking some time of your busy schedule to be with us today and having this very interesting and very important conversation. We are looking forward to hearing more news about your developments. I don't know, maybe we will see you in Basel, Switzerland in a month or so.
No, I
planning
don't.
to go there.
I don't get there, but hopefully in the next coming symposium or meeting, some congress on ALS. Hopefully, yes.
Okay. Nevertheless,
Okay.
Thank you for working on ALS on a regular basis. Thank you for keep fighting. against ALS, and we in the ALS Association of Argentina, we have all been affected by ALS. Some of us are relatives to ALS patients, like myself. I lost my brother to ALS, unfortunately, and some others that are part of our association are patients, and they are still here with us. So again, thank you so much, and we're looking forward to hear more about your work and to seeing you again.
Yes, thank you so much for your invitation, and we all keep the fight. Thank you so much.
Of course. Thank you, Dr. Devos. Bye.
Thank
Take
you.
care.
Bye-bye. Bye-bye. Take care.
Thank you.